Hello everyone, hope you're doing well. Today we're going to be taking a look at the process of cellular respiration within the cells. Quick note before we get started, this is a presentation based off of the IB International Baccalaureate definition of cellular respiration, and therefore we will be mostly utilizing International Baccalaureate's biology textbook as our resource. So first we'll start with what is cellular respiration. Essentially, respiration is the controlled release of energy from organic compounds within cells. Typically, it involves breaking down carbohydrates, but it can also occur with lipids and proteins. So primarily the cell is going to prioritize breaking down carbohydrates, but it can also utilize lipids and proteins. The cell creates energy via the form of adenosine triphosphate, or ATP. That's essentially the molecule that is used as energy for various functions within the cell. Respiration can be aerobic, meaning it uses oxygen in the mitochondria, or it can be anaerobic, which does not require oxygen or the mitochondria. But the downside to that is essentially that anaerobic respiration yields much less ATP. So before we get started with the actual process, let's take a look at how ATP is used. It's made up of 3 phosphate groups together as one, and triphosphate. When it's hydrolyzed, or when water is added, one phosphate group detaches, and this reaction releases energy. The leftover ADP, adenosine diphosphate, di meaning 2, is reused to create new ATP later on, but that's not part of this process quite yet. ATP is used all over the body for various functions because, many of these functions require energy in order to work, hence the importance of cellular respiration. All right, now, real quick, let's take a look at the carrier proteins as well. So respiration utilizes these different carrier proteins in order to add and remove, and also transport protons and electrons. Most commonly H plus or hydrogen. Most common carrier is NAD plus, and when it is reduced to add hydrogen, it becomes NADH. FAD is also present, but less common, and it forms FADH2 when H plus is added. Okay, there's a little diagram there of how that works. Now, with all that out of the way, we can get into the actual process. So cellular respiration begins with glycolysis. Glyco meaning sugar, lysis meaning breaking apart. So glycolysis occurs in the cytoplasm, and it does not require oxygen in order to create ATP. Hence, this is the part that's anaerobic respiration. It makes significantly less ATP than the Krebs cycle or the electron transport chain, but its advantage is the fact that it does not require oxygen. So the process begins with a hexose sugar, so 6 sugar together, and it becomes split into 2 triose molecules. Oxidation then occurs, removing hydrogen from those triose sugars, as you guys can see here on the diagram on the left. That hydrogen that's removed reduces the NAD plus to become NADH. As we said before, those are going to be important for later. And then as a result, 4 ATP molecules are formed, 2 of which are going to be used to continue the process in the Krebs cycle, and the other 2 are the net yield. On top of that, we're going to take out the 2 leftover pyruvate molecules, and they're going to be broken down during the link reaction in the next stage in the mitochondrial matrix. Okay, now for the link reaction. That's kind of the second part, but essentially the 2 pyruvate molecules that were taken from the end of glycolysis enter the inner layer of the mitochondria called the mitochondrial matrix. Enzymes then remove one carbon dioxide and one hydrogen molecule taken by NAD, and it becomes NADH, obviously. And this is called oxidative decarboxylation. Oxidative meaning oxidation is the removal process, and carbon dioxide and the removal of carbon dioxide, that's decarboxylation, hence oxidative decarboxylation. This creates one acetyl group that will react with coenzyme A for each pyruvate molecule, so 2 acetyl-CoA are produced, in addition to NADH. Okay, now for the next stage, the Krebs cycle. It starts with the 2 acetyl-CoA, which are 2C molecules, entering the Krebs cycle, and they join a 4C sugar molecule to create a 6C molecule. Oxidative decarboxylation occurs, which removes CO2, leaving 5C, and then it happens again, leaving 4C. At the same time, hydrogen molecules are absorbed by NAD and FAD, and those become NADH and FADH2 respectively. The 5C to 4C reaction that also joins ADP with another phosphate group, creating one ATP molecule, and it's worth noting that's per each pyruvate molecule, so that's going to be 2 ATP. As a result, there's NADH and FADH2 to carry out the electrons, and 1 ATP molecule per pyruvate, meaning there's really double of each. CO2 is a waste product, and it's removed from the cells when you exhale, when you breathe out, actually. So that's a waste product. And here's a diagram to show you what I'm saying. At the top, you can see the acetyl-CoA, and it joins with the 4C to become citrate, which is the 6C molecule. Then the carrier proteins take away the carbon to make it carbon dioxide, and take the hydrogen, the H+, to make it NADH. Then it happens again, back to a 4C. ATP is produced, and then FADH2 also takes the leftover H+ ions, and we're left with a 4C molecule, which starts the process all over again. But essentially, our products are the different carrier proteins and the 2 ATP. Okay, now for the final part of the process, which is the electron transport chain and chemiosmosis. So the electron transport chain is located in the inner mitochondrial membrane. That's kind of the space between the wall and the central part, which is the mitochondrial matrix. So all these NADH and FADH2 that we've collected from glycolysis, the link reaction, and the Krebs cycle, these essentially are going to come over to the mitochondrial membrane, the inner membrane, and they are going to kind of drop off their hydrogen ions. So the hydrogen ions are going to move over to the other side of the inner membrane via passive transport, and the electrons are going to move in the inner membrane space, kind of between, in the middle of the membrane, and they're going to activate the other channels to allow more H+ ions to pass through. And this process is called oxidative phosphorylation. Once there's a high enough concentration of H+ ions on the inner membrane, they begin to move through the hydrogen carrier in the membrane, and that generates the highest amount of ATP from each stage, about 32. So as you can see, aerobic respiration, this process, generates significantly more ATP than would otherwise be created from anaerobic. And any of those excess H+ ions are absorbed by oxygen, which creates H2O, or water, as a byproduct. So we'll take a look at this real quick. So as you can see, in the inner membrane, the H plus ions flow through the carrier through the membrane proteins, and the electrons activate those proteins. They move up to the inner membrane space, and then flow back out, creating ATP and H2O. So, just to recap here, all the products that come from cellular respiration. At the end of the process, there's going to be roughly, like meaning it's going to vary, but there will be roughly 6 CO2, 36 ATP, that's 2 ATP from glycolysis, 2 from the Krebs cycle, and 32 from the electron transport chain, and there will be roughly 6 H2O produced. Those hydrogen carriers, after they drop off, it's worth noting, the hydrogen carriers, after they drop off the H plus ions, are going to move back to the beginning of the cycle and start all over again. But yeah, that's pretty much the net of the of cellular respiration. Hope you guys enjoyed. Hope that you learned something from this. Thanks for watching. Take care.